Hey guys, and today we're going to talk about the lens transparency. So let me write the topic here. Lens transparency. Okay, so isn't this amazing? You know why? Because amidst the complexities inside the human body, and despite the fact that the whole of the human body is overrun by 5 to 6 liters of blood, the lens, the just small structure that is about 9 to 10 millimeter of diameter, that much small organ or small structure that remains untouched, that's not even stained by a single drop of blood or any kind of fluid. This is really amazing thing. The human body is so much, in a way, organized, body or structure. So what are the features or what are the regions that helps the lens to remain transparent? So we're going to talk about that. So the physical factors that play a significant role in keeping the lens transparent, crystal clear, are: number one is avascularity. Yes, the lens is the avascular part of the eye. Okay, lens is not supplied by any arteries, neither it is drained by any venous system. So you may be wondering how it gets nutrients then. Well, it draws nutrients from the aqueous humor and a little bit nutrients from even the vitreous humor. Okay, from the vitreous humor, and maximum of its nutrition from the aqueous humor. Okay, so number two reason for lens transparency is the role of the lens proteins. Okay, lens has proteins, and basically many books says these proteins fall under the aquaporin superfamily, and these are further classified as aquaporin MIP 26 or aquaporin 0. These are the proteins that fall under the superfamily aquaporin. So the presence of these proteins helps the lens to maintain its transparency. Okay, and the third reason is pump mechanism. What actually this pump mechanism does is the pump mechanism of the lens membrane. It helps in electrolyte and the water balance inside the lens, so it keeps the lens hydrated all the time. Okay, and the fourth reason is lens capsule. As I already told in the previous lecture, that lens capsule is a transparent hyaline membrane structure. Yes, it is transparent. It is rather semi-permeable. Okay? Semi-permeable, transparent, and hyaline membrane. So it also helps the lens to retain its transparency. And the number fifth reason is the autooxidation and high concentration of the glutathione. Yes, not oxidized glutathione, rather high concentration of reduced glutathione. Okay, And yes, these are the things. And oh yes, and I should not miss another thing, that is the characteristic of the lens fibers. The lens fibers are, these are so compactly arranged. They are so compactly arranged. Had this been, arranged this way, then it would have decreased the transparency of the lens. All right, but since these are arranged so compactly, there is not even a very small space between these fibers, so this helps in maintaining the transparency of the lens. Okay, so the next next characteristic of the lens fibers are, it is very narrow, very narrow, very thin. And the next next characteristic of the lens fibers are, it doesn't have organelles. Okay, as I already told in the previous video, these lens fibers are nothing but the elongated columnar epithelium in the equatorial region or the posterior portion of the lens, right? So when the epithelium gets matured and gets elongated, they lose the nucleus-like organelles, right? Which play a role in not maintaining the transparency of the lens. So when these organelles goes off from the cell, this also helps in maintaining the transparency of the lens. So these are the basic 6 reasons or 6 factors that helps in maintaining the lens transparency. So these all points are clear to you, I guess, but I think I should give much time, a little bit more time in this 5th point. That is, I hope you're seeing this. Let me see, you're seeing, yes, you're seeing this 5th point, auto-oxidation and high concentration of glutathione. So let me get into more detail about this point. Before going into this point, I'd like to tell a little bit about the free radicals. What are free radicals? Okay, you know what? About 1 to 4% of the oxygen we take are converted into free radicals in the human body. Okay, we take oxygen, right? When you take oxygen, this oxygen goes inside the body, and this oxygen is converted, 1 to 4% of the oxygen is converted into free radicals. Free radicals. And these free radicals, yes, first question is, what are free radicals? Okay, free radicals are nothing but these are the molecules or molecular fragments that contains one or more unpaired electrons, one or more unpaired electron in the outer orbitals. Okay, and these free radicals or the reactive oxygen species are, for example, superoxide ion, superoxide, that is represented as like this, and nitric oxide and hydroxyl radical, which is represented as this, and even S2O2. Okay, even S2O2, right? But actually this S2O2, S2O2 and this O2 singlet, I hope you're seeing this. Let me check out whether you're seeing this or not. Okay, you're seeing, cool. Actually, this S2O2, hydrogen peroxide, and O2 singlet, oxygen are not considered as the free radicals. But actually, since these are so much reactive, so these are also included in the, in this group. Okay, but actually, in real scenario, these are not actually the free radicals. But since they have so much reactivity with the molecules of the human body, so these are also grouped as the free radicals. Yes, anyways, let me turn this page. And these free radicals, when these free radicals, let me align this paper correctly. Yes, cool. When this free radical react with a normal compound, the free radical, suppose this peroxide, when this react with the normal compound, this causes a regeneration of more free radicals. Okay, more free radicals, and this chain reaction leads to thousands of events. And suppose this is your cell and this is your cell membrane. Okay, the cell membrane has a major component that is PUFA, that is polyunsaturated fatty acids. So when the free radicals come and react with this PUFA, this causes disruption of the membrane, right? This causes loss of function of the cell membrane, and if it is lens, it causes and this, if this is a protein, this is lens protein, aquaporins. When the free radicals react with the protein, then it causes disruption of the protein, and this causes the transparency of the lens to decrease. Okay, this decreases the transparency of the lens. Okay, so these free radicals, these free radicals have to be disposed. These free radicals have to be destroyed so that these free radicals won't damage our normal human body structure, normal lens structure, normal proteins, right? So human body is so much organized, it has even some prophylactic mechanisms, even preventive mechanisms. If this happens, what should be done? That has already been programmed in the human body. So human body is really amazing, So there is a system called free radical scavenger system. This system is nothing but this system is a combination of the enzymes like superoxide dismutase enzyme, glutathione peroxidase enzyme, and glutathione reductase enzyme, and catalase, and like this. What these enzymes do actually is these enzymes help in destruction of the free radicals, and they make sure that these free radicals are converted into inert compound water in most of the cases. Right, so let me describe the reaction that takes place while destroying the free radicals. So suppose this is your superoxide free radical. When this superoxide free radical reacts with H plus, it forms hydrogen peroxide, which is also a very destructive molecule. Okay, and oxygen also formed, I guess, and this reaction occurs in presence of superoxide dismutase. Right, now the important steps comes now onwards. The hydrogen peroxide in presence of glutathione peroxidase enzyme is converted into H2O water, which is very inert compound, right? And for this reaction to happen, there is a need of very important compound, that is glutathione, GSS. We write in short GSS, or glutathione. Hydrogen peroxide in presence of glutathione peroxidase, it changes into H2O2, which is very inert compound, that is water, and this takes place in presence of glutathione, rather reduced glutathione. And after this reaction, the reduced glutathione is converted into oxidized glutathione. Okay, and this oxidized glutathione has to be again converted into reduced glutathione so that this reaction is continuously occurring. Okay, so that it makes sure that this reaction continuously occurs in the lens, and the free radicals do not get a single time to destroy all the normal structure of the lens. So for the conversion of oxidized glutathione into reduced glutathione, there should be an enzyme called glutathione reductase. Glutathione reductase. This glutathione reductase, in presence of NADPH, converts the oxidized glutathione into reduced glutathione. And here comes only NADP plus. And you may be wondering from where this NADPH comes. The one and only reliable source for NADP is nothing but HMP, SUNT pathway. You might have been known about this pathway. You might have studied in your basic science level. HMP SUNT pathway is the major source of NADPH in the human body. Okay, the HMP SUNT pathway takes place in presence of glucose 6-phosphate glucose 6-phosphate enzyme. Glucose 6 rather dehydrogenase, glucose 6 phosphate dehydrogenase enzyme helps in formation of the NADPH, and this NADPH in presence of glutathione reductase changes the oxidized glutathione into reduced glutathione, and this reduced glutathione in presence of glutathione peroxidase enzyme converts the dangerous peroxide ion, dangerous hydrogen peroxide, into inert compound water. So this mechanism is continuously occurring in the lens. That's why there is no any disruption of the proteins in the lens, and this helps in maintaining the transparency of the lens.